However, we're going to do the best that we can to our ability to have a fun event. The first speaker is our very own faculty advisor, Dr. Han. She does some amazing work with flies, and she's going to talk about, I'm sorry?
I'm sorry, that was on my end. Go ahead.
She does some amazing things with flies. If you guys were keeping up on our social media, one of the posts that we had displayed, the graphics, the fly graphics, it had shown some of her work. It had to do with giving alcohol to some of the flies, and what you found was that they were more, they had less inhibitions during the alcohol intake, which was very interesting, and it was mentioned at a very prestigious article. For those, I'm going to keep it like a little bit below the science. I don't want to, I know some undergraduates don't exactly know what it is to publish, and you probably all don't really know how we do research using the fly model. So hopefully once Dr. Han gets started, you guys can learn a little bit more about that. So we would like to thank you, Dr. Han, for dedicating all your time, being an advisor, and basically mentoring us professionally. And she's basically the one that has been helping us uh, organize this event. So we'd love to have you, Dr. Han, and I'll let you take it away so that you can basically show everyone what it is to be a neuroscientist. Thank you.
Great, thank you, Ree. It is my genuine pleasure to have this chance to talk to you all. And I'm so looking forward to hearing what you're thinking about this as well. Well, it will be good for me to talk about how to, or what it is like to be a neuroscientist. I think I need to set aside time again because I prepare my talk to chat a little bit about what we do in the lab in our research in a way that you can have better understanding on how we use an animal model like fruit flies to understand the brain. So that's why the title is more like a Flies Are Us. I don't know, have you guys heard of Toys R Us? The company bankrupt. Nonetheless, I think you guys have fresh memory about it, although it's 10 years down the road, 20 years down the road, nobody will remember what it is. But my goal today is to give you an idea how we use animal model to gain more insight into kind of mechanism. Mechanism may be something new to you, in an easy way to understand the pathways, how the brain tend to process information that we receive from the environment and process it to provide the way that you react or remember or do whatever you do in your everyday life that include rest and sleep and eat and poop and everything they can think about. So almost everything is under the control of nervous system. What I hope to focus on is the brain. So with that one, I will go ahead and share the PowerPoint slide with you. But I just want to make sure that you understand what I hope to do. One, please turn on your video so I can see you. Two, you can type up questions that you have during my talk, so I can try to have some communication feel as opposed to me giving a talk to the audience beyond cyberspace. So let's try to make this more fun experience for you as well too, right? So that would be, I think, the most important goal of all that we hope to achieve today. And I do understand that obviously not everybody has background in neuroscience. And therefore, what I try to do is to explain the terminology as much as possible. So let me go ahead and have my chat box show up because right now after I do this, chat box disappears. So I have a chat box and then I have a everybody in gallery, and I have my talk. So organization. So I think now we are all in a good kind of starting point. So once again, I already made a small kind of introduction on how the brain mediates the information that we receive in the environment that can be good or bad to trigger learned experience or learning that we say or remember things that we experienced, as well as giving us kind of way of controlling our thoughts and behavior that allow us to execute everyday life. Because as you think about, even driving from your home to the campus require quite a bit of control in a way that while you're driving, obviously you need to know where to go to be able to do so. You need to be on, for example, I-10, and you need to know where to get off, right? So all you need to remember. And sometimes you do it without thinking too much out of it, because a lot of things that you do repetitively become habitual. So you don't need to utilize conscious effort. But pretty much everything that we do is a mixture of unconscious and conscious efforts of the experience that we gain through our kind of experience of the environment and what we do. Obviously, they require a lot of obviously molecules in the brain. The particular molecule that we focus on is known as monoamines. Some of you who know or who are having depression may be familiar with monoamine, because people with depression tend to take monoamine oxidase inhibitors. So like tricyclics, tetracyclics, that way people know monoamines in that way. Monoamines include, for example, serotonin that's linked to depression, as well as dopamine and norepinephrine. So we pretty much focus on monoamine system in our brain. Recently, we've been studying acetylcholine, which is another major neurotransmitter in the brain. So our goal is to figure out how these molecules actually are mediating information that we acquire to provide adequate outcome in our behavior, in our thoughts. So with that, let me just go ahead and give you a bit of a kind of introduction to our lab. So as I told you, the monoamines acetylcholine, they are known as action neuromodulator. I don't want to get into details on terminology, but these are key signal in the brain. I think there will be a messenger and the receiver. So these are so-called messengers. And then we are studying this messenger system because they are critical for learning and memory and drug abuse and addiction. We do use alcohol as a main drug of abuse and addiction that I will describe why later. And also beyond these behavioral changes that we call behavioral plasticity, we are also studying how we control our inhibition. Well, if we want to do everything that we do, then, our life becomes totally chaotic. We know when to stop eating, we know when to stop playing and study. Those are all inhibitory control, very important cognitive inhibition. But also there are many important kind of non-conscious, unconscious inhibitory control that we have. So I will go ahead and talk about how we do so. Although I'm not going to talk at all today, one of our study is on female reproduction, female ovulation, because the molecule that we are working, which is dopamine norepinephrine system, is critical for controlling ovulation in female. This is fly abdomen and this is human abdomen. You can see already there's a huge kind of similarity even in anatomy. So we believe there's some common mechanism underlying ovulation mediated by adrenergic system. So that's one area that we are working, but that will be something beyond our scope because this is neuroscience talk, and this will be more cell biology, neuroendocrine system, which is neuroscience, but the study that we do is more in oviduct, fallopian tubule, as opposed to nervous system. Recently, we have started working on aging and dementia that you shall see why, because research is something that you can set up the goal. But sometimes while you're doing research, you find something really exciting that guide you to open up a new area of research. That's how research go on forever, more or less. It's not like you're done on one project. Oh, I'm done with the research. Now it's time for me to do something else. It ain't gonna happen. It always happens in a way that we try to look into mechanism for something that discovers something totally unexpected. Unexpected discovery tend to be, I think, a lot more significant in many ways than targeted, because targeted research and finding tend to be within our capacity to imagine. But nature and science is much beyond what we can imagine. So when there's anything that is spectacular, that is beyond what you hypothesize, now leads to even bigger discovery about science. So I think that in that way, this is definitely something that we are really excited about. As I discussed earlier, even in the title, we do all this in Drosophila melanogaster. So everybody already know what Drosophila melanogaster, based on the picture that you can see. However, there are many different types of flies, and this particular fly species is obviously fruit flies. So why flies? Why fruit flies? So I should say why fruit flies, because there are house flies as well. Oh, I always do this fruit flies. What's good about them? Well, obviously, this is a traditional genetic model, meaning their genome is fully sequenced, and there may be a mutant for almost every gene in the fly genome. So you can now take a look at all the fly mutant. So if you want to know the function of a gene X, how do you do that? Well, you get rid of X that we call mutation, you mutate it, and compare the animals without mutation and animals with mutation. And whenever there's difference between these two groups, we conclude that obviously whatever being different is the one done by the gene that is being mutated. It makes sense. That's very simple logic. And this is typically how people tend to use genetics to understand the function of individual genes. So obviously there are many genetic variants in human population as well too. As you know, we humans have about 3 billion nucleotides, base pairs. In the case of flies, they have 180, which is about how many times less? Those who are good at mathematics are doing calculation already. 10 times, much more than 10 times, right? 20 times, less than 20 times. The exact number would be 16.6. So let me go ahead and say 17 times less. Humans. So that's why it is a lot easier for us to understand individual genes in the fly genome than in human genome, because they're a whole lot less. Well, why are there a whole lot more in humans? Obviously, humans are a lot more advanced. So there are many genes that are unique to humans, but also they tend to have multiple genes to do the same thing as one gene does for flies, that we call functional redundancy. So flies do not have much functional redundancy. So when we mutate one gene, we can see the consequence right away, which may not be the case in the case of humans. Well, on top of that, you cannot artificially mutate human population. You're gonna be jailed if you do it, right? So people do this in monkeys and, you know, a little higher kind of animal model like rats and mice, but still they have some functional redundancy, which is much less in flies, which is wonderful. the most important maybe. If fly genome is totally different than humans, who cares, right? But the matter of fact is a lot of genes in fly genome are conserved in humans, or the other way around. A lot of human genes are conserved in flies. So there are a lot of conservation in terms of individual genes too. So a lot of genes that we know for disease like autism, Parkinson's disease, Alzheimer's disease, you name any disease, there are fly counterparts. And fly has been serving to understand how those mediated genes tend to trigger the kind of symptoms that people have, and also help us to understand how we go about develop therapeutics as well too. So in a way that this is really important. And maybe I can go a lot faster from now on, where flies do have a short life cycle. Life cycle meaning male and female mate and get a progeny for it to become full adult. And in the case of humans, God knows, it takes, well, for fertilization to a baby being born, it takes 10 months. But in the case of flies, it takes 12 days. It's less than 2 weeks. How about lifespan? Obviously, some flies live short. Well, in wild, maybe they die earlier because they don't get good nutrition. And they get cut by humans and die on site. But in the lab, if it provide good food, they can live over 2 months, which is a long time for us to study aging. So this is another very important. And if flies are really stupid, then they're not going to be able to be useful for the study of learning and memory, right? I mean, they're not as sophisticated as humans, but they're not as, in some cases, dumb as some humans. So that may not be a good way of saying it. So they do have certain intellectual capacity that are supported by the central brain highlighted in here. I'm not going to go through details out of it, but we do focus on this particular brain structure named mushroom bodies because their structure looks like a mushroom. And the function of this mushroom body may be similar to hippocampus, which is really critical for long-term memory formation in humans. So this will be kind of one short introduction on the brain, and I'm not going to get back to how this works. But instead of talking about individual brain structure, I thought maybe good for me to talk just a little bit about how brain work. How brain work? It worked through multiple cells are communicating a lot of information at the same time. So imagine when you think of a messenger and a receiver, if you think of FedEx guy who's driving around to deliver the package, right? So he's so-called messenger, neurotransmitter. When you're at home and you're receiving it, you will be the receptor, right? So that's how package is being delivered. This is how the information is processed in the brain. Well, for FedEx guy, it typically happens at the door of your house. In the brain, it happens at the particular structure. Does anybody know this particular structure is named? Communication unit in the brain. This is pre and this is post. Somebody's typing up 2 new messages. Yes, synapse. Emily, excellent. Sibasi, excellent. If there's a way that you can give them a little gift, that would be wonderful. Brianna. This is known as synapse, right? In the case of a human brain, does anybody know how many neurons that we have? Neurons are the cells that are communicating in the brain. Go away. The name of the cells in your skin, epithelial cells. The cell in your muscle cell, right? neurons that are communicating in the brain is named neurons. How many neurons do we have in our brain? 86 billion. Yes, Isabella. Did you take a neurobiology class? Excellent. 2 thumbs up. So then now 86 billion is a lot of cells in the brain, right? And most of them are born when you're born. So you have them till you die. So they are long-lived. I mean, there are new places that are 2 places in the brain that are making new cells, but when you think of the numbers, minority compared to 86 billion. I'm not going to talk about neurogenesis in the brain. As you experience more, you get more neurons, obviously. But individual neurons are communicating with many other cells through synapse, right? In average, how many synapses per neuron? 10 synapses, 100 synapses, 1,000 synapses, 10,000 synapses. How many? I guess you guys never won any lottery. 10, 100, 1,000, 10,000. Pick one. Nobody want to bet money? Well, you're not even betting money. Too good, too good. Anyway, 1,000. Excellent, Nico. Nico is another word, if you can see, right? So with that one, 1,000. Some have less, some have a whole lot more. Obviously, Purkinje cells has 100,000. I think that's how many synapses that Purkinje cells in the cerebellum has. Anyway, there are a lot. So when you think of a total number of synapses, it will be 86 trillion. That's a lot of synapses. I think this number alone represents how complex our brain is, right? So when one cell gets information, gets a lot of information at the same time. So understanding what's happening at the synapse is really important for us to know how individual cells or pathways, circuitry tend to engage in mediating different types of brain functions. So for it, the key element, neurotransmitters, they are messengers. Receptors, they're very important for receiving information that modify the output of the cell activity. And this unit is known as synapse. I think that is very important information. So with that one, obviously, our focus here will be dopamine. How many of you have heard of dopamine previously? I hope most. Because this is really famous molecule. Why? Well, this is linked to addiction. This is linked to ADHD. It is linked to schizophrenia. It is linked to Parkinson's disease, right? And it is linked to also autism as well. It does many different things because in regular brain, it tends to mediate many brain functions that include attention, emotion, motivation, so many things. And this tiny dopamine molecule is actually made from amino acid tyrosine. Then you may wonder how this tiny molecule can do so many different things. Right? That's legitimate question. The way it does is by having many different receptors in many different brain areas. So this is a really good schematic presentation of dopamine neuron and postsynaptic neuron. So as I told you earlier, maybe I'm talking too much, I don't know. Let me go ahead and cool off a little bit. So pretty much dopamine is made in presynaptic dopamine neurons packaged in the synaptic vesicle. When the cell gets information, the synaptic vesicle fuse to the membrane to spit out dopamine, which bind to the receptor in postsynaptic site that modify neural activity that mediate pleasure, for example, that mediate motivation when you do a lot of different things. And then the excess dopamine need to be adequately taken back into presynaptic site so the information cannot be just going out forever, right? So this particular molecule known as dopamine transporter is really important for timely regulation of dopamine information to the postsynaptic site. The significance of dopamine transporter is really well documented by cocaine. Everybody knows what cocaine is? Yes, obviously. Did you know that side of cocaine's action is dopamine transporter? So that's it is. So cocaine, when people take cocaine, which is not a good thing to do, obviously, it blocks dopamine transporter, thereby increasing the level of dopamine in synaptic cleft. So it keeps on activating receptors, leading to enhanced signal. So instead of having happy, then people who are taking cocaine say this euphoria, which is a lot more heightened pleasure. And that kind of activity modify neuron activity that make people experiencing that one. Not everybody, obviously. Some people keep on seeking cocaine. So that is so-called addiction, right? So key point that I made here, as you may have noticed, not everybody who take cocaine become addicted. There are only a subset who tend to get addicted. So a lot of other factors involved in addiction beyond initial response to the pleasure or euphoria. So we try to understand how that happened, how, not just increasing pleasure, but how taking a drug like alcohol does the same thing, by the way, indirectly working on dopamine system. So how different kind of receptors tend to be important for alcohol abuse and addiction as well too. So with that, I think, by now, we need to introduce, okay, so much on the background. So how about how we actually use flies to understand the mechanism, so-called mechanism by which dopamine mediate different types of brain functions and how abnormality in those functions tend to lead to addiction or learning and memory problem. So to study that, we need to have some kind of way to measure it, right? So the way that we measure is by looking at behavior. Behavior is ultimate outcome of the brain activity. So behavior is one that we use the most in our lab, in addition to immunostaining and molecular analysis like RT-PCR. So looking at the assays, behavioral assays that we are looking at, I just made a list of some behavioral assays that we do. So for simple learning and memory, we utilize olfactory conditioning, which is passive learning and memory paradigm, as you shall see. Another paradigm that we use is condition courtship. So let me just go ahead and give you a kind of overview on how these two learning and memory assay work. To be able to do it, I think I need to use animation. So I'll go ahead and put it in kind of this way. So I show one fly here, but there are a whole bunch of flies in a kind of container. In this case, a tube. While flies are moving around, we give them an order with electric shock, which is painful. Trust me, it is really painful. And obviously, electric shock, painful, they're not gonna like it. So this is a really important kind of way that we can measure how well that they remember. So even in the absence of electric shock, since that electric shock came when they smell channel number 5, for example. Now, whenever they smell channel number 5, they avoid it. This is in a way similar that, let's say, you met somebody robbery, for example, in the street of Oak Street. I know it's not in there because I don't want you to make any associations. I made up the street name. So while you're walking down the street on Oak Street, you met a person with a gun saying, "Freeze, give me everything that you want." So you got robbed. Your next response, you never go there, right? Because now Oak Street gives you bad memory and therefore you avoid it. So in a way, this aversive conditioning is the same, where flies tend to remember this smell to remember bad odor. So this is a really good way for them to remember. But in other case, let's say while they are smelling channel number 5, now they get banana. Well, we use sugar. Actually, they love sugar. Then this is good experience, right? Then whenever they smell channel number 5, now they get attracted to it. So the cue is the same. With one experience, they got electric shock. With another experience that they got banana, sugar. So this is a really good way for us to map the brain pathway or mechanism, how bad memory versus good memories tend to be formed and maintained. So with that, obviously, we use this one in the level last. So in one of the newest actually paper done mainly by Martin, who was undergraduate student here at UTEP, done most of work in collaboration with the postdoc, Paul Sabando. So he identified actually adrenergic receptor beta is really important because when we imitate them, they do not learn this aversive conditioning, and we can rescue this deficit by putting it back into machine bodies. So that is recently published in Journal of Neuroscience. But prior to it, we identified many other players playing really important role in the machine bodies. So I thought that this is one of the typical example of how we can identify the mutant in the gene of interest. Once we identify the mutant, we can identify this mutant by using RT-PCR to show that they make a whole lot less transcript and therefore they make less protein. So when we utilize them, this is quantification. When we utilize them in our olfactory conditioning, compared to the wild type, they do not learn at all. So this shows very nice kind of paradigm. And this is once again, as I told you earlier, to learn this kind of learning and memory, flies don't need to do anything, right? So we place them in a container, give them electric shock with odor. It's really passive. What you're doing right now could be passive if you have to come here, but I don't think it's passive because you voluntarily came here. So I think it is a more active learning experience for you. Then how do we measure active learning? That is by using so-called condition curve chip. So in this case, we are using flies' natural behavior. In particular, male. In flies, I don't think that's the case in humans anymore. Males tend to be a lot more aggressive in sexual behavior compared to female. So this is male. So when male sees female, he quotes her and then copulate, which is all great. However, when a male see mated female, he still quote her. However, mated female is laying eggs, so she kicks him and runs away. So this is a really active rejection, just like, you know, some guy that you don't like come after you and they say, no, I don't want to date with you. You do it, right? It is active rejection. So this guy who got rejected by a mated female don't like that experience. Who would, right? You can easily imagine. So what he does is that even with virgin female, he kind of remember bad experience and therefore he stopped coding for a while. So this is active learning and memory in a way that he needs to code female to learn. Yeah, some females can be really not to do it. I try not to use a word that may not be good for me to say in public talk, but everybody understood what I tried to say in here, right? So he becomes a little more kind of selective next time. So this is a really important paradigm for us to study active learning and memory. So in our study done by actually June, who used to be research associate, now she is at Emory University, and master student Anna had done was to look at many different receptor mutant. So this is typical, this is without rejection, and this is with rejection. So you can see with rejection, they code whole lot less. So simply this is looking at courtship index, how much time did they spend on coding female. So when we look at all the mutants, like T1 mutant, T5 mutant, DER mutant, they tend to show really good learning. Except for this one particular receptor mutant, they do not remember at all. So on our after, a lot of mutants as well as wild types to remember rejection, and they do not code as much. But this mutant just forget right away and they start coding. So this is actually really distinctive kind of a receptor in many ways because it binds to steroid receptor that may be really critical for us to understand how our brain tend to have dopamine and estrogen or progesterone tend to work together to mediate sexual behavior. So this is one study that we are pursuing in that regard. But more importantly, I think this is a really key finding that June made, which is that T1 receptor is really important for motivation. So when one think of sexual behavior, there could be many different kinds. Well, I should not say that. There are many different partners, right? They're not all the same. There will be some appealing partner, less appealing partner. So if one is really motivated, then one may not be selective in terms of coding. This is without rejection. This is native kind of courtship activity. So in the case of wild type males, they tend to code just a regular female as well as the female without head. So we tend to decapitate female to make them less active so we can measure male sexual activity more. But in the case of Cantonese wild type, they don't care whether female is intact or decapitated. But as you can see here, the flies that is deficient in T1 tend to show much less with decapitated than intact, meaning that T1 receptor is really important for sexual motivation. And then we map that function in the mushroom body. As you can see here in the mutant, they do not have T1 in the mushroom body at all compared to wild type. Now we can exogenously put T1 only in subpopulation of mushroom body structure, and then we can rescue the phenotype. This is published in G2B, so I'm not going to go into details, but this is typical genetic manipulations that we do to understand how different dopamine receptors tend to engage in different learning and memory processes and components in the learning and memory as well too. Question about this so far? Before I go into more exciting part, I think, which is alcohol. Pretty much you guys are in legal age for drinking. And I'm sure many of you are drinking, correct? So obviously, the paradigm that I discussed so far is the learning and memory paradigm that we use to compare, natural stimulus-induced changes in behavior. But a lot of drugs, including alcohol, tend to modify behavior. And drug addiction may be better understood as enhanced memory about the cues associated with drug, yet dampened memory about all the others. So there will be imbalance in terms of how animals tend to make and maintain memory, which tend to be more geared toward drug-associated cues. So with this one, obviously we use alcohol because we used to use cocaine. And since flies don't want to eat cocaine, we volatilize the cocaine So when we volatilize cocaine, it comes out. So students tend to get exposed to cocaine. That typically worries me very much. So I was not sure whether that's a really good way to recruit undergraduate students to work in the lab. So we changed the drug to something that is legal. At least alcohol is legal. And this is something that a lot of people tend to get exposed to. So as you may be able to appreciate, alcohol does a lot of things. When you drink, you become hyper. Hyperactivity can be sometimes a really good representation of euphoria, happiness. Just like children, when they get happy, what do? They run around, right? So hyperactivity could be one representation of euphoria. And when you drink a lot, then it make you pass out, sedation. Also, as you can imagine, alcohol trigger disinhibition, making some people use all kinds of four-letter words, make some people become more aggressive, making some people be disinhibited. So a person may be shy because there's a lot of inhibition going on. And with drinking, they become all of a sudden more talkative. Or when it comes down to sexual advances, they tend to be more open and more risky in their sexual activity as well too, right? So, and there are also a lot of adaptive changes, as you can see here. This is the first, kind of time for flies to get exposed to alcohol. With time, so this is time and percent flies that are stated. So with time, there are more and more flies as stated. On their first exposure, like on your 18th birthday, I don't think that's the case, but when they drink for the second time, as you can imagine, it takes much longer for them to get stated. This is typical process that we call tolerance. So there's a lot of studies done on this avenue, but our focus is more on sensitization, which is known to be linked to closely drug addiction, which is heightened behavior. So one good example out of it, this is one that I use all the time in my talk. Now my students are using it. So whenever you do dishes at home, so today you get $5 when you do dishes. Tomorrow when you do dishes, you get $10. Next day you do dishes, get $15. Likelihood that you do dishes next day, high. So when people do drugs, the euphoria that they feel is a lot more heightened each time. So they tend to seek more alcohol or cocaine. So this is one phenomenon known as disinhibition, and this is one phenomenon known as behavioral sensitization. So we thought that this is a really good avenue to explore with flies using sexual behavior, because obviously sexual behavior needs to be done as a group. So this is typical way how actually in the absence of flies, when you put a whole bunch of males together, this is what they show. Nothing is happening once in a while, but this fly become horny and then try to coat the other male, and this male rejects him and runs away. So there's very little activity going on. However, when so this is actually, which is different from the male-female courtship. So as you can see, when male sees female, he pursues her actively and then sing a song by flipping wings that we call courtship song. And then he licks her genitalia. And then when she likes him, then she slows down and then he succeeds in copulation. This is a ritual that typically happens in fly sexual behavior. So when you have a bunch of flies together, obviously, when you give them alcohol multiple times, that's what's happening. So this is courtship song, courtship chain, where, can you see the singing? Can't you see sometimes licking? So you have an idea regarding how flies become sexually disinhibited in a way that they tend to court the other male. That doesn't happen on the first exposure, as you can see in this video. On the first exposure to alcohol, they are just busy, they become hyper, but you see very little intermale courtship behavior. But when flies get alcohol once a day, every day, the level of intermale courtship tend to be enhanced. That is typical definition of behavior sensitization. So this is one way that we study how dopamine system tend to engage in courtship sensitization, which we have published in multiple papers. So this is one study. Actually, it is already 5:51, so I'm gonna go a little faster because I think this is even more exciting in my opinion. So additional kind of a study that we do regarding dopamine is to understand how dopamine tend to be important for behavior control and how abnormal dopamine tend to underlie impulsivity. So impulsivity is inability to suppress the behavior that's not proper under the circumstances. So although people know that they should not smoke on campus, you see some people out of impulsivity or something they do. Although you're not supposed to eat dessert because we are full, you're still eating. Well, there are many other impulsivity that you can think of. So we use fly as a model system to better understand actually impulsivity and inhibitory control. This is typical kind of measurement in humans to look at inhibitory control. So in the case of ADHD study, human subjects are supposed to press the button when they see alphabet. P, press button, K, press button. Whenever they see X, they need to hold it. So A, press, X, hold, B, press, X, hold. So one has to really pay attention to the task and hold their movement really well. So as you can see, children with ADHD make a lot more mistake when they do this Gonoga test. So we made kind of a fly version for Gonoga test. And then the way it works is that, we kind of let flies move around in a chamber. And then we give them blow, which is kind of strong wind that we have today outside. When it happens, what do? You stop to make sure, right? That everything is you're not going to be blown away. Obviously, you're not going to blow away. But this is typical reaction to the wind, and which is pretty well done in wild type animals. As you can see, they hold their behavior really well. But when it comes down to, so this is quantification. When it comes down to kind of neurotransmitters that is responsible for it, we mutated a lot of neurotransmitters to see which one is important. This is when we manipulate actually a particularly dopamine system, which is adrenergic system. This is strong, under the influence of strong strong wind. Instead of stopping, they decide to court the other guys. In these guys, instead of stopping, they tend to show grouping behavior. So by manipulating transmitter system, we can modify their output. One study that we are pursuing right now is that when we give a strong airflow, wild type flies are stopping really well, but as you can see here, this is the mutant flies defective in dopamine transporter. They cannot hold their movement, they fly around. So this is what we call, so to speak, impulsivity or loss of inhibitory control. And exciting part of it is that when we look at a single fly in a chamber, they do really well. However, when we put more and more flies in the chamber, as you can see with time, is number of flies that are showing flying behavior, impulsivity. There are more and more flies showing impulsivity at earlier onset. So from here, we learned that social factor is really key in interacting with the hyperdopamine. So just in a way that, how people who are doing drugs like cocaine, under certain circumstances, tend to become a lot more aggressive, in a way. So that may be a good example that you can correlate. So in our lab, we try to understand how that happens by looking at the pathway at the synapse and by looking at the circuitry that I'm not going to talk too much out of it. But we are also using this finding to identify additional molecules, so-called genetic screen. Due to time, I'm not going to go through details out of it. The idea behind it is that we tend to lower kind of heterozygous instead of homozygous mutation and look for another heterozygous mutation that trigger impulsivity. So when we did that screen, I apologize for going really fast, but I don't want to go beyond too long because I am getting hungry. I'm sure you're getting hungry too. So we don't want to pass out because we are too hungry. I feel like I'm passing out. So we had done genetic screen, which is typically what people do when they study Drosophila, because that's why Drosophila is really beneficial to look for something unknown gene critical for inhibitory control. So one gene they identify that are critical for enhancing impulsivity is known as SCALI. Have you guys seen X-File? It's from X-File. So obviously a person who identified this gene function named it SCALI. So there are a whole bunch of related names, but SCALI is one. So what is it? Obviously it's a mitochondria protein. Does a lot of many things, but one thing that caught our eyes, it is the one that binds to amyloid beta. Are you familiar with amyloid beta? Yes. This is the one that is accumulating in the brain of people with which disease? Which disease? Oh, I wrote it down. God, stupid. Alzheimer's disease, right? So this is where we got into studying aging. So we identify SCALI being important for loss of inhibitory control interacting with fumin, which is dopamine transporter, in young age. But now we looked at just SCALI mutation alone. And guess what? So in young animals, they are fine. But as they get older, now they show more and more loss of inhibitory control. So SCALI is critical for aging sensitive inhibitory control. As you can see from your parents, maybe your parents are not that old, maybe your grandparents. I'm getting that stage, unfortunately. They're not good at inhibitory control, right? They are saying things that are not supposed to be proper. They do things that are not supposed to be proper. So as we get older, there will be kind of overall loss of cognitive function. But when it comes down to disease state, which is known as dementia, they tend to be a whole lot more augmented. So that augmented behavior is shown in here as a impulsivity under the influence of a strong air. So we are looking into now how these genes that are critical for aging sensitive inhibitory control in acetylcholine pathway, because acetylcholine is one that is known to be important in cognition with aging, which is the one that is most affected by Alzheimer's disease. So and then also the enzyme that breaks down is known as ACE. And then ACE inhibitor is the first line medication for treating Alzheimer's disease and dementia people. So this is the hot kind of data that several people in the lab had worked together. So it will be Pat, Paul, Aldo, I think those are main players, working on how acetylcholine system tend to respond to aging. As you can see here, young animals, this is a distribution pattern for the enzyme that is making acetylcholine or transporter. In mid-age, there's dampening. Then flies don't stay dampening because there is a loss of acetylcholine possibly. Now they boost up the enzyme to make more. So there's adaptive change at the level of the enzyme. So when we look at obviously Gonogo test with aging, now they show loss of inhibitory control that is dampened by mutation in ACE. The same is true for learning and memory as well, but what we learn even more exciting, this may have some impact in human translational potential. In the absence of dementia, actually, in normal animals, by mutating ACE, just like treating them with Aricept, they have a whole lot better cognition. So I think maybe overall, enhanced acetylcholine may support enhanced learning and memory system as well. So this is one avenue that several members are working really hard that I highlighted. Maybe, no, I didn't highlight them. Working really actively. So I think I had gone through a lot of behavioral essays, but the bottom line is that by using many different learning and memory essays, we try to dissect, for example, how dopamine, this tiny molecule, does so many things in the brain, through which we tend to we try to understand how not only gene and environment tend to work together along with the social kind of factors, work together to trigger abnormal behavior that may have some biomedical implication. So with that, I think I'm going to stop with the acknowledging a lot of work done by the postdoc in the lab, Dr. Sabando. Without him, I don't think we'll make this much achievements. And then there had been a lot of students coming in and out, but Eric Saldes is PhD student who's about to finish, and Patricia is a bioengineering working in our lab along with the Dr. Boland. And then Aldo, I think he will be starting PhD program in hopefully fall. And Abraham is master student, and Jose is undergraduate student, Rice Scholar. Dillian is actually Emeritus Scholar. And Adrian is from EPCC. So these individuals are ones who are still coming to work in the lab, which is wonderful. This is the lab picture taken 2019 December, last time, because we could not take a picture in spring semester 2020. So it's kind of sad to share this because there's no 2020 spring, no 2020 fall, but hopefully we can have one in 2021 spring and for sure 2021 fall. So with that, I'll stop and take questions. You have any questions?
Alex, you might want to unmute everyone else so that they can start asking questions, or if you guys want to drop it in the chat box. Alex, I noticed that you put question. Did you have a question?
Yeah, but I can ask. I think she already answered it. So I'll go ahead and ask everyone to unmute, and you don't have to unmute, but if you have a question, you can go ahead and use that as the time to unmute. So go ahead now, if anyone has any questions.
This is new to everybody. You should not be shy. So no, you should not have any inhibition when you ask a question. Oh, I did not do a good job conveying information.
That
was a really
wasn't
good
it at all.
presentation.
You do a lot of interesting science. I hope everyone got the chance to understand a little bit more about how we relate fly brains to like human science. Did you have a question, Brian? Sorry.
Yes. Brian? You can unmute first.
Okay. I had a question. So dopamine increases cocaine increases the dopamine inside the fruit flies. Is there any cases where it doesn't have an effect on fruit flies? like where it doesn't like have an addiction effect?
So cocaine is known to, it is really well studied molecule regarding target site. So cocaine binds to dopamine transporter in flies and humans and any animal model that you look at. So that's the main target site. It also binds to serotonin transporter. So there will be some differences in maybe efficacy on how it or binding. So I'm not sure whether there's a huge difference in vitro, but there are a lot of studies done regarding biochemistry in vitro. In vivo is harder to know which is which. But in terms of in vitro study, can bind to serotonin transporter, so thereby increasing serotonin. But its major action on addiction is through more dopamine. If you mutate dopamine transporter, obviously, it's not going to respond to, obviously, cocaine. But at the same time, when people study animal model, mutating dopamine transporter also trigger different types of outcomes. So some animals with mutation in dopamine transporter show certain aspect of addiction as well too. So dopamine transporter may not be the only target site that lead to addiction. There may be additional targets that does the job, and maybe norepinephrine transporter could be one. Serotonin transporter, increasing norepinephrine or increasing serotonin, those could be one potential mechanism as well too. Does it answer your question? But in the case of humans, maybe you were kind of thinking of why not everybody who are taking cocaine don't become addicted to it, right? So in that case, there are multiple factors. One, obviously, cocaine will get metabolized inside the body. So one, how fast it will get metabolized. So there will be pharmacokinetics, like alcohol. You know, me, I cannot drink alcohol because I cannot break down aldehyde. So alcohol become aldehyde, alcohol break down, so you get rid of alcohol. So like my sister, I've never seen her drunk. I've never seen her face being red, but she drinks a lot. She metabolizes alcohol really fast, but I cannot metabolize aldehyde. So I become really red and get rashes. I need to scratch everywhere. And then when people look at my eyes, I look like Dracula. So that's me. So I'm pharmacokinetically inadequate to metabolize alcohol aldehyde. The same is true for cocaine. Some people tend to metabolize right away, so its effect tend to be short-lived, so it may not. In other people, maybe the residence time can be longer. Some people may be more kind of activated dopamine system, so they are likely to go through a lot of adaptive changes. So initial take of cocaine does not do a whole lot, but is more repetitive intake, slowly changing brain activity, making people sick for the cocaine. So when it comes down to addiction, there are multiple reasons multiple pathways, how people become addicted. It's not one pathway, it's multiple. So in that, people tend to have different activity in dopamine systems through their, not only genetics, but also their prior life experiences as well. So our brain activity is unique in a way that, as I told you, there are 86 trillion synapses, and those activities are unique to you, just like social security number. Not the same individuals having the same synaptic activities in your brain, they're all unique. So depending on your genetic makeup, depending on your life experience, those synaptic activities tend to be different. So the way that you learn the information from this talk in the class, also the way that you respond to cocaine or alcohol tend to be different. So there will be a lot of heterogeneity. And also, obviously, experience that you have when you drink alone versus with your friends tend to be quite different. You agree on that one? right? Environmental setting of your drinking. I cannot use cocaine as an example, so I'm using alcohol as an example. You experience the differential effect of drinking with different setting, meaning the way that alcohol works in your brain tend to interact with many other factors as well too. So it's not really simple. That's why the brain is so complex. At the same time, that's why it is so exciting to study it.
Thank you.
Does it
There was
answer your
some.
question, Brian?
Sorry, so yeah, so it was great answer. Thank you very much.
Okay, wonderful. Any other question?
Yes, Dr. Han, there's some questions in the chat.
Uh-huh.
One
So-
person is asking if there are any requirements to enroll in research for fall.
Oh, so there's no requirement based on President Wilson. 100% classes, including research credit, will be face-to-face. You know, I love to have students in the lab. Always the limitation was getting permit from the school. So there has been a lot of regulation regarding, you know, there needs to be 2 people in the lab, no more than 3 people at the same time, during rotation and stuff like that. I think they will go away in fall. So there's no requirement. If you want to do research through, for example, research credit, do not hesitate to contact me. And yes, typically I do not screen students. I believe equal opportunity for everybody to get engaged in research. And that's why FLY is a really good model system. Not everybody has that kind of a liberty of doing it because rats and mice are expensive, but flies are cheap. So we can have as many students to come and enjoy what they can do for us.
Yes,
So
that was a really good question because actually a lot of the students, I believe the last questions in a prior meeting, they asked how to get into a research lab. So maybe you want to give them a little bit more of feedback on that since you're, you know, the head of the lab and you decide who joins or not.
So what you need to do is to look up faculty webpage. We used to have an individual webpage, but now we have more kind of centralized. So what they show is kind of expertise connector. So when you press faculty name, it goes to expertise connector. And then if you get in there and then search for the faculty name, it will show the page with their expertise. And then if the topic is of interest to you, then you can email, dear Dr. Han, I looked up your information on expertise connector. The study topic on inhibitor control sounds really exciting to me. I'd like to gain hands-on experience in research. Would it be possible for me to take research credit in your lab in fall semester? So just don't send group email to all faculty. Do you have a slot? Can I take your research credit? So you need to do your share, which is to look up the information, showing your interest in doing something meaningful, and therefore I'm sending you email. So I get sometimes email like, can I take research credit with you? Just that simple. Then, you know, those students do not put in any thoughts into what they hope to do. I mean, there are some students who are desperate to get 1 credit before graduation, which I understand that too. But I think proper adequate way of doing it is to look up research profile of faculty in the subject. If you want to study cancer, then, you know, just go to research connector and then put cancer, and they will bring up the list of faculty who are working on cancer based on keyword search. Then go ahead and take a look at what they are working on, and then contact the faculty member. In some cases, faculty may not reply to your email right away, possibly because sometimes they are busy and then they just forgot to reply. But if you send the email again, you know, for me, serious students, motivated students may email twice at least. So whenever I get email again, then I reply right away. So just don't give up, and then don't say, oh, I did my best by sending an email, but she did not reply to my email. Everybody is busy, and then it's always good to give a benefit of doubt. Oh, maybe my email did not make it. So it's always good to follow up. I think good professionals are really good at follow up. When you do good follow up, a lot of things that you desire will happen. But you do this and then don't see it coming to the way that you hope. You give up, nothing's gonna happen. So I think follow up is a really important thing to do. Always follow up. What to lose? It's only a couple of minutes of your time. You just send the email again. I sent you email on March 12th, but I did not hear from you, and I wonder whether my email got to you. So I'm sending you this email again. So it's very simple. So I think having a good follow-up is a really good thing to do. And whenever you do things proactively with, proper etiquette, you will hear from faculty. So a lot of faculty members are open in their kind of options of having students starting on research credit first, and then if you do well with it, you may stay in the lab as a kind of you can pursue scholarship. We have a lot of undergraduate scholarships, including RISE program, CRE program, and BUILD program. So there are a lot of opportunities. You may be able to do research with financial support as well too. And if you really like what you do, then you can pursue graduate school here or elsewhere. But if you want to get into really good graduate program, then gaining undergraduate research experience and productivity is really important. So that would be something that you have to see by doing research. Sometimes, research is not for everybody. So in that case, you need to give it a shot, really, without knowing how it is like. It is something that you get excited about. I mean, excitement is one. If you feel like, oh, I need to do it, it's not a good one. I want to do it. So you need to figure out, I need to do it or I want to do it. When you think you want to do it, then you can get into research.
Yes, I completely agree. That's why most of us are all here. We love talking about science. Dr. Han, we're a little bit over time, and there were some other questions, but maybe if you guys could email Dr. Han.
Yes.
Yeah, would that work? I'm gonna put her email in here. And we do have another speaker in the next okay, Alex, can you reply to Brian?
Yeah, social press. Yeah, I got you.
Thank you guys so much. And thank you, Dr. Han. That was okay, I see where you're at. That was extremely well. I also attached her research article on Nature, which for any undergraduates, I really suggest reading this because the actual publication might be a little bit complicated. But always try to, look for opportunities. And even though research isn't for everyone, you got to give it a chance, and you'll definitely get a better sense of what you want to do for the future. I know we're all still kind of unsure, and I know a lot is going on in the world right now where we can't participate in person, but that doesn't mean that you shouldn't at least try once, right? So, all right, then I'm going to go ahead and stop the presentation so we can get ready for the next one. So if any of you guys need to take up those links, go ahead and do so now.
Kitaki. So it was really nice talking to you all. And then do not hesitate to contact me if you have questions or if you want to chat. And if you want to do research, obviously, in fall semester, then definitely let me know as well, too. Right?
Yes,
So
that was a great offer.
Great day.
Bye,
Thank you.
Dr. Han.
See you around. Bye.